Howdy science enthusiasts! Today we'll extract DNA from two vegetables I bought from the grocery store. First, the spinach, and secondly, the onion. DNA stands for deoxyribonucleic acid. It is the code of life. In plants, the DNA is located mostly in the nucleus, but is also found in mitochondria and chloroplasts. Let's first talk about the materials that we will need. We will need a bunch of things including isopropanol, iodized or non-iodized salt, water, laundry detergent or dishwashing liquid, mortar pestle, sieve, measuring cylinder, and straw or spoon. Let's get started with the process of this DIY DNA extraction protocol. This is 70% isopropyl alcohol or isopropanol. I have already poured this in a plastic cup and I am now keeping it in the freezer at minus 20 degrees Celsius. I will take this tap water and pour this in this plastic cup. Now I am adding iodized salt. I am stirring it with a straw to dissolve the salt. Now the salt is dissolved in water. This is a laundry detergent. I'm adding this to the salt water mixture that we just prepared. I'm mixing this gently with a straw and I'm trying to avoid bubble formation as much as I can. Now, the solution containing salt and laundry detergent is ready for downstream applications. Let's call this LS solution. I will now grind the samples. I'm using the mortar and pestle that I have at home to grind the sample. I already ground the spinach leaf and now I will pass it through the sieve. I am placing this sieve over a wide mouth plastic cup and pressing the ground leaf sample from the top. To this collected sap, I am now adding LS solution. Similarly, for onion, I will take 2 pieces of fleshy leaves or scales. I am now crossing them as described previously for spinach. The onion scales are now ground. I am now passing them through the sieve and collecting the sap in a new wide mouth plastic cup. Also, to this collected onion sap, I am now adding LS solution. Similarly, we will also keep a negative control to strengthen our experiment. In this third wide mouth plastic cup, I will add just the LS solution without any plant material. We will gently swirl all these 3 plastic cups to mix the components. Now, to each of the 3 plastic cups, I am adding cold isopropanol that I had earlier kept in the freezer. I will give a gentle swirl to each of the 3 cups. Now we will wait for 5 minutes to see the results. The wait is over, and now we can observe what we got. One thing to note beforehand is that DNA does not dissolve in alcohol. First, the spinach leaf. Here you can see sticky mucus-like threads in the suspension. Those are aggregates of DNA in spinach. Let's also look at the onion sample. Here also we have thick, sticky strands of DNA. Observe the white mucus-like substance sticking to the straw. Now, let's finally locate out negative control that did not contain any plant sample. There's nothing in here. What just happened? Let's talk about the underlying principle here. What did the grinding do? It broke down the cell wall and released cell components in the suspension. What did the salt do? It dissolved protein. It made DNA less soluble in water. What did the laundry detergent do? It disintegrated or dissolved cell membrane as well as the nuclear membrane. This led to the release of DNA from the nucleus. And what was the role of cold isopropanol? It increased the precipitation rate of DNA. Thank you so much for watching. Please like, share, and subscribe.